The global public health community is currently assessing reports of a significant genomic development regarding the hantavirus, a pathogen already known for its severe impact on human respiratory systems. Scientific teams involved in genomic surveillance have identified a specific mutation within the viral sequence that may correlate with an accelerated onset of clinical symptoms, specifically those targeting pulmonary function. To understand the gravity of this development, it is necessary to examine the foundational nature of hantaviruses. These are a family of viruses spread mainly by rodents, and in the Americas, they are the primary cause of Hantavirus Pulmonary Syndrome, or HPS. The disease is characterized by a high mortality rate, often cited in medical literature as being between 35 and 40%. While the headline suggests a discovery that instantly shuts down human lungs, medical reality usually involves a progression from initial prodromal symptoms to a critical phase of respiratory failure. The significance of identifying a mutation lies in how the virus interacts with human biology. Normally, hantaviruses are categorized as zoonotic, meaning they jump from animals to humans but rarely spread between people, with the notable exception of the Andes virus strain in South America. The reporting of a new mutation raises immediate questions for clinicians and epidemiologists regarding whether the viral fitness or the mode of transmission has been altered. For viewers and the general public, the reason for concern is the rapid nature of HPS. Once the leakage phase begins, where blood vessels in the lungs begin to spill fluid into the alveolar spaces, the window for medical intervention narrows significantly. This report frames the current understanding of these genomic shifts against the backdrop of existing public health protocols. We are looking at a pathogen that does not require a complex vector like a mosquito. Instead, it utilizes the everyday environment of rodent habitats. When scientists speak of a genome mystery or a new mutation, they are often looking at the protein spikes on the viral envelope which dictate how easily the virus enters a human cell. If a mutation makes this entry more efficient, the resulting viral load can overwhelm the immune system before a robust defense can be mounted. This sets the stage for our technical look at the core developments within the laboratories and the specific mechanisms of the virus that are currently under the microscope. The core of this development centers on the structural biology of the hantavirus and the specific genomic segments that dictate its behavior. Hantaviruses are negative-sense RNA viruses with a genome divided into three distinct segments: large, medium, and small. The large segment encodes the viral polymerase, the small segment encodes the nucleocapsid protein, and the medium segment encodes the envelope glycoproteins known as GN and GC. It is within these glycoproteins that many critical mutations occur, as they are responsible for cell attachment and entry. Scientists investigating the current mutation are likely focused on how these proteins bind to integrins on the surface of human endothelial cells. When the virus successfully hijacks these cells, it triggers a massive inflammatory response. This is not a traditional infection where the virus destroys cells directly. Rather, it is the body's overreaction, a cytokine storm, that causes the capillaries in the lungs to become permeable. The source materials indicate that this specific mutation may be linked to a more aggressive shutdown of pulmonary capacity. In a clinical setting, this manifests as Hantavirus Pulmonary Syndrome. Patients initially present with fever, myalgia, and fatigue, which are indistinguishable from many other viral illnesses. However, the transition to the cardiopulmonary phase can be abrupt. Official statements from public health agencies emphasize that laboratory confirmation is essential, typically utilizing polymerase chain reaction, or PCR, to detect viral RNA or serological testing to find specific antibodies. In specialized laboratories, often requiring biosafety level 3 or 4 containment, researchers are attempting to sequence these new variants to determine if the mutation is a localized event or a sign of broader viral evolution. There is a clear distinction between verified scientific data and the more sensational claims of an instant shutdown. While the biological process of capillary leak syndrome is rapid, it follows a path of viral replication and immune signaling. The mystery mentioned by researchers often involves viral reassortment, which occurs when two different strains of a virus infect the same host cell and exchange genomic segments. This can lead to sudden shifts in how the virus behaves in a human host. Current surveillance data from health ministries suggests that while cases remain sporadic, the intensity of the respiratory distress observed in recent patients has prompted this deeper look into the viral genome. Labs are currently working to replicate the mutation in controlled environments to see if it increases the viral fitness or the speed at which it can disable lung tissue. This technical work is the front line of defense, as understanding the mutation is the only way to eventually develop targeted therapies or improve the accuracy of diagnostic kits used in rural clinics. Prevention and public health surveillance remain the most effective tools in managing hantavirus risks, especially as new mutations are identified. Public health guidance remains consistent. The primary route of infection is through the inhalation of aerosolized virus particles from the saliva, urine, or droppings of infected rodents. When people clean out cabins, sheds, or storage areas where rodents have nested, the act of sweeping or vacuuming can kick these particles into the air. If the reported mutation does indeed increase the virulence of the virus, the margin for error in these settings becomes even smaller. Health officials recommend a wet cleaning method. Instead of sweeping, individuals should use a disinfectant or a mixture of bleach and water to saturate any contaminated areas, ensuring the dust is not kicked up. Regarding the limits of current knowledge, there is no specific vaccine or cure for hantavirus that is widely available in many regions. Treatment is primarily supportive, involving the use of oxygen therapy and, in severe cases, extracorporeal membrane oxygenation, or ECMO, which breathes for the patient while their lungs attempt to recover. The uncertainty surrounding this new mutation lies in whether current diagnostic tests will still catch the virus or if the genomic shift is significant enough to evade detection. Furthermore, it is unknown if this mutation affects the environmental stability of the virus, how long it can survive in the open air before losing its ability to infect. Public health agencies are calling for increased surveillance of rodent populations, particularly in areas where human-rodent interface is high. This involves trapping and testing rodents to see if the mutation is widespread in the animal reservoir or if it was a rare jump to a human host. The calm but firm advice from the medical community is to maintain rigorous rodent control and to seek medical attention immediately if a high fever and muscle aches follow any potential exposure to rodent-infested areas. As scientists continue to decode the genome mystery mentioned in the latest reports, the focus remains on early detection and hospital-based supportive care. The situation serves as a reminder of the constant evolution of zoonotic diseases and the need for robust, global genomic tracking systems. For further updates on public health developments and viral research, please consider subscribing to this channel. The investigation into the reported genomic mutation of the hantavirus involves a rigorous examination of the viral structure, specifically focusing on how the pathogen interacts with the human pulmonary system at a molecular level. At the center of this inquiry is the hantavirus genome, which is composed of 3 single-stranded RNA segments, the large, medium, and small segments. The medium segment, or M segment, is of particular interest to the scientific community as it encodes the viral envelope glycoproteins, known as GN and GC. These proteins are the primary tools the virus uses to bind to receptors on the surface of human cells, specifically the beta-3 integrins found on the endothelial cells that line the blood vessels within the lungs. A mutation in this area, as suggested by the recent genomic surveillance, could theoretically alter the binding affinity or the speed at which the virus enters the host cell. While the phrase instantly shuts down lungs implies an immediate cessation of function upon exposure, virologists and clinicians describe a more complex, albeit rapid, biological progression. The reported mutation appears to influence the transition from the initial viral replication to the acute phase of Hantavirus Pulmonary Syndrome, or HPS. In this stage, the body's own immune response, rather than the virus itself, causes the most significant physiological damage. This phenomenon, often referred to as a cytokine storm, leads to a massive increase in vascular permeability. As the integrity of the blood vessels fails, fluid leaks directly into the alveoli. The tiny air sacs in the lungs, effectively preventing oxygen exchange and causing the patient to experience severe respiratory distress. The core development in current research is determining if this mutation accelerates this leakage phase or increases the viral load to a point that overwhelms medical intervention more rapidly than previously documented strains. Medical facilities and diagnostic laboratories are currently on high alert for cases that exhibit this accelerated clinical path. Testing for hantavirus remains a specialized and time-sensitive process, typically involving two main approaches. The first is molecular testing via reverse transcription polymerase chain reaction, or RT-PCR, which identifies the presence of the viral RNA in the blood or tissue samples. The second is serological testing to identify the presence of specific IgM and IgG antibodies, which indicate an active or recent immune response to the virus. Because hantavirus is classified as a high-consequence pathogen, work involving live virus must be conducted in Biosafety Level 3 or Biosafety Level 4 laboratories. These facilities are designed with high-efficiency particulate air filtration and strict decontamination protocols to prevent any accidental release. Scientists in these high-containment labs are comparing the newly identified mutated sequences against established global databases to determine the origin of the genomic shift. They are also investigating whether the mutation is a result of antigenic drift, which occurs through gradual mutations, or viral reassortment, a more sudden change that happens when two different hantavirus strains exchange genetic material within a single host cell. This distinction is critical for public health agencies as they assess the risk of a more widespread or potent outbreak. Current reports from hospitals indicate that the window for successful medical intervention may be narrowing. While there are no specific antiviral medications currently proven to cure hantavirus, the primary clinical development involves aggressive supportive care in an intensive care unit. This includes the use of mechanical ventilation and, in the most critical cases, extracorporeal membrane oxygenation, or ECMO. This life support system bypasses the patient's lungs to provide oxygen directly to the blood, giving the pulmonary tissue time to recover from the massive inflammatory event. The contrast between sensationalized reports and the clinical reality remains a point of focus for health officials. While the term instant may be hyperbolic in a literal sense, the speed of the pulmonary crash in patients, often occurring over just a few hours, is a well-documented characteristic of the disease that this newly discovered mutation may be exacerbating. As researchers work to decode the genomic shift within the hantavirus, the focus for public health agencies shifts from the laboratory to the front lines of prevention and environmental surveillance. Because there is currently no broadly available vaccine or specific antiviral therapy for Hantavirus Pulmonary Syndrome, the primary defense remains the interruption of the transmission cycle. This cycle is fundamentally zoonotic, relying on the presence of rodent reservoirs such as the deer mouse, the white-footed mouse, or the cotton rat. Public health guidance emphasizes that transmission occurs almost exclusively through the inhalation of aerosolized viral particles. These particles are shed in the urine, saliva, and feces of infected rodents and become airborne when disturbed. In light of the reported mutation, which may correlate with a faster clinical progression, the necessity of rigorous environmental hygiene is heightened. Practical takeaways for the public centre on the concept of wet cleaning to prevent the aerosolization of dust. Individuals are advised never to sweep or vacuum areas where rodent activity is evident, such as cabins, sheds, or garages that have been closed for the season. Instead, officials recommend saturating the area with a disinfectant, specifically a mixture of 1 part bleach to 9 parts water, or a commercial disinfectant labelled as effective against viruses. A dwell time of at least 5 minutes is necessary to ensure the viral envelope is chemically deactivated before the waste is wiped away with paper towels or rags. For those working in high-risk environments, the use of personal protective equipment, including non-porous gloves and N95 respirators, provides a critical barrier against the inhalation of contaminated particles. Beyond individual action, global surveillance programs are intensifying their monitoring of rodent populations. This One Health approach recognizes that human health is inextricably linked to animal health and environmental factors. Surveillance involves the systematic trapping and biological sampling of rodents to determine the prevalence of the virus within a specific region. If the newly identified mutation is found to be widespread in the animal reservoir, it signals a potential shift in the regional risk profile, requiring updated alerts for local clinicians and emergency departments. However, there are significant limits to current knowledge. It is not yet clear if the mutation affects the environmental stability of the virus, or if it alters the traditional rodent-to-human transmission path. There is also the question of whether current diagnostic assays, which are programmed to look for specific genetic markers, will remain 100% effective if the virus continues to evolve. Ethically, the challenge for public health officials is to communicate the gravity of these genomic developments without inducing unnecessary panic. While the reported mutation is a serious concern for the scientific community, the virus still requires a specific set of environmental conditions to infect a human host. The ethical closing of this report must highlight